Hello everyone, so this is Practical 3, Physical Properties of Different Lipids and Fatty Acids. So the introduction of each member will be in the next video.
Hi, my name is Stb1083. Hi, my name is Shafayudina.
Hi, my name is Zulaika.
Hi, my name is Victoria Jesse.
So this is the introduction of the physical properties of different lipids and fatty acids. Next, we have 4 objectives of the experiment. The first objective is to understand the correlation between the physical structure of lipid products to the physical properties of lipids and fatty acids. Next, to determine the solubility of lipids in polar and non-polar solvents. The third one is to identify the appearance of various lipid products. The last one is to determine the relationship between the solubility of lipid products to the physical properties of lipids and fatty acids. Let's move on to the materials and experimental procedure. Alright, as we can see here, we use 2 kinds of material, which is microcentrifuge tubes and also dropper. So the function of microcentrifuge tubes is to hold and make small amounts of lipid-containing food samples and solvents. For the dropper, we use for dispense small controlled drops of liquid lipid samples and also solvents. Based on the methodology, we have to put 5 drops of each lipid-containing product into the tubes. The third material used is distilled water. It acts as a polar solvent in the solubility test. It helps demonstrate that lipids do not dissolve well in polar substance, emphasizing their hydrophobic nature. In step 4, the first set was begun. Approximately 1.4 ml of water was added to each centrifuge tubes using a pipette. We started adding the distilled water into the microcentrifuge tube containing olive oil. Second lipid-based food, which is ghee. And the last one, the shortening. Based on the manual, we also used tube spatula, but it wasn't shown here because it was prepared beforehand. The function of the spatula is used to transfer solid lipid samples into the microcentrifuge tubes with precision and hygiene. The tubes were kept and shaken to mix. So this is the outcome of first set after mixing with water. Alright, for the second set, we used a toluene as a non-polar solvent, and the toluene was added into each sample. The toluene here acts as the non-polar solvent in the solubility test. It is used to show that lipids are soluble in non-polar substance. The pipette tips was disposed to avoid contaminants. Let's move on to the second one, which is ghee. The same step was repeated. We also use the pipette tips to mix the lipids and also the toluene, so the process will be much better and easier. The observation of the mixture were recorded. So this is the outcome from set 1 and also set 2. So this is the result. As we can see here that lipid is insoluble in polar solvent, meanwhile soluble in non-polar solvent. The conclusion of the experiment. Lastly, reflection based on experiment 3. The first one, new learning. Through this experiment, we gain a better understanding of how lipids behave in different solvents. We learned that lipids are generally insoluble in polar solvent but are soluble in non-polar solvent. Next, we also became more familiar with identifying lipid-containing substances in everyday food products, which help deepen our awareness of the biochemical composition. Lastly, we also enhance our ability to handle the lab equipment such as microcentrifuge and also droppers accurately. Second reflection is challenge phase. One of the main challenges we face is during observation. Some samples had weak odors, which make it difficult to compare them effectively. Lastly, when it comes to toluene, we were a bit concerned at first because of its strong fumes and potential hazards, but we managed to handle it safely by doing it in fume hood and use face masks. Last but not least, suggestion for improvement. Firstly, a clearer explanation on how to observe and record solubility such as layering, cloudiness, or dissolution will make the experiment more consistent across the groups.